Great. Hi, everyone, and welcome to our first seminar of 2024. Just for those of you who may be new to the seminar, my name is Amanda, and I will be the chair for the seminar today. So feel free to ask your questions in the YouTube chat at any point throughout the talk, and we will relay them to the speaker live during our question and answer period at the end. And so just a quick introduction to our speaker today. We welcome Dr. Elinor Lichtenberg as our first speaker of 2024. So Dr. Lichtenberg completed her PhD research at the University of California, San Diego in the Department of Biological Sciences. Her PhD research focused on foraging and dominance behavior in stingless bees. And after her PhD research, she completed a series of postdoctoral research positions across the USA before settling at the University of North Texas, at which she is currently an associate professor as well as head of her own research lab in the Department of Biological Sciences. So her current research focuses on understanding plant-forager interactions, insect foraging, as well as insect biodiversity. So today she will be giving a great talk with a really good title entitled Eavesdroppers and Robbers and Spies, oh my, Decision-Making by Foraging Bees. So thank you again, Elinor, and you have the floor.
Hi, everybody. Thanks for coming to this talk. I love the promotion. I'm actually still an assistant professor. So associate professor, hopefully in a few years. So today I'm going to talk to you about some of my research that deals with bee foraging behavior. So in my lab, we're broadly interested in how various types of human activities are altering plant and animal biodiversity, foraging behavior, and ecological functioning. And to do this, we integrate behavior and ecology, and this lets us address some questions that are at the interface of these two disciplines. And overall, we've got two goals in the lab. So one is to advance our fundamental understanding of behavior and ecology related to these questions. And then the other is to use this science to improve management and conservation. And we focus largely on flower visiting insects. So this is things like bees and butterflies, but also various predatory and parasitoid insects. And so these predators and parasitoids actually will come to flowers. It turns out that they can increase their reproductive output by drinking nectar and getting the sugars from that nectar. So lots of different things at flowers, but with a major emphasis on bees, especially for the behavior component of our work. And so bees, they show some really interesting foraging and social behaviors. So for example, bumblebees can solve the traveling salesman problem to get the most efficient route among a set of flowers. Bees are really good at learning to associate various types of information with food availability. They also, on the social side, the social bees show really interesting division of labor, conflict over reproduction. So they're a great system for looking at, especially for looking at foraging behavior, which is the focus of my research. And then they also are really important for regulating ecological processes and for providing benefits to people. So we think that about 90% of the flowering plants worldwide and 75% of crops benefit from insect pollination. And then these predatory insects, they are providing pest control. They're eating the herbivores, and so benefiting both crops and also natural systems. And both pollination and pest control then are having really important benefits for us people in terms of things like human food production and nature for us to enjoy. And we think that these groups contribute hundreds of billions of dollars per year to the global economy. So they're really important from an economic viewpoint as well. And so in my lab, we're addressing three broad questions. So we look at community ecology, asking how various types of land management affect habitat, and then biodiversity and interactions that animals have. And then we look at behavioral ecology, which is what I'll be focusing on today, looking at insect foraging behavior and also a little bit at cognition. And we also look then at pollination biology, so biodiversity, and then also behavior. What do these mean for plant reproduction? How are they affecting pollination? And so, like I said, I'll be focusing mainly on the behavior research today. So a key theme of my research is interactions. And from an ecological viewpoint, these interactions are very important because they link various components, and they can really help us understand some patterns that we see between habitat, biodiversity, and then ecosystem function and services. It's really the interactions that are the mechanisms bringing about these patterns. But they haven't been a big part of the conversation in this part of ecology yet. But of course, interactions is largely what animal behavior is all about. And so from a behavior perspective, we have a heavy emphasis on interactions that can help us better understand ecology. And we also know on the behavior side that interactions, they're not just having these ecological outcomes, but they're really important for altering fitness of the individuals that are interacting. And so today I'm going to be talking about interactions within the context of foraging. And so foraging, or finding food, for us people, it's pretty easy. You go to the grocery store, but for something like this bee, there are a lot of different challenges that she may face in efficiently finding food. So things like deciding what types of food to eat, figuring out what locations currently have food, which individual flowers have food, which in this case is nectar, and how to handle flowers to extract the food out of them. And interactions can help overcome these challenges. And I'm going to talk today about two different types of interactions. So an individual can interact with their food, or they can interact with other foragers. And I'm going to address these through three case studies. So first, I'll talk to you about nectar robbing bumblebees and how they use alternative food handling tactics as they interact with flowers. And then I will talk to you about social information and how it can help animals learn about food availability. First, I'll talk about eavesdropping stingless bees and then spying bumblebees. So we'll start by talking about nectar robbing bees and alternative food handling tactics. So when an animal comes up to some food, there may be more than one way in which she can handle it and extract the food or make it available for consumption. So for example, if we look at this wasp here, she can take her prey item whole back to the nest, or she can cut it up into little bits and carry those bits back to the nest. And then on the other side of the screen, we see an example with predatory snails that drill in to these bivalve shells to eat out the animal that's living in there. And so they can drill in either through the wall or through the edge as two different food handling tactics. And you'll sometimes see that one individual is capable of handling food items in more than one way. And so, of course, that raises the question of, well, why does that individual use certain tactics in specific situations? So, for example, with these Eurasian kestrels, they bring prey back to the nest for their chicks to eat. And with smaller prey, like lizards and insects, they just bring them back whole. But when they have larger prey, like large voles, they will sometimes decapitate them, presumably to reduce the weight that they have to fly with as they're carrying it. So we do see this flexible use of different tactics. And the specific food handling tactic that I'm going to focus on today is nectar robbing. So when a bee or other flower visitor, including hummingbirds, comes to a long tubular flower, sometimes she will cut a hole or use an existing hole at the base of the flower and drink nectar out of that hole. And we call that nectar robbing, and it's an alternative to sort of visiting at the front of the flower. So I will be talking today about three different ways that bees and other animals can handle flowers. So the first is legitimate visitation. And this is where the bee comes to the front of the flower, goes in through the flower opening, and is probably acting as a pollinator. But then we also see robbing, where she's making a hole or going to those existing holes and drinking nectar from those holes. And we can divide this into primary robbing, where the bee is making the hole herself, and secondary robbing, where she's using an existing hole. And so you can see here, this bee's robbing up on this flower. And then throughout the talk, I'll be using this icon for legitimate visitation and this icon for nectar robbing. And so nectar robbing has been studied largely from the perspectives of community ecology and the evolution of mutualisms. And it is seen there as exploitation of the pollination mutualism. So these robbers, they're getting nectar from the plant, but they are not in turn providing reproduction or pollination to the plants. And so they're seen as exploiting the pollination mutualism. And so ecologists have been very interested in this behavior in this context. But they have sort of reached a point where they're a bit, there's a big question that's been raised. So if you look at mutualism theory, it assumes that there's a cost to being a mutualist, which in this case is legitimate visitation. And so therefore there should always be selection to exploit the mutualism and show the exploitative behavior, which in this case is nectar robbing. And so there's a prediction that if an individual is capable of nectar robbing, it will always nectar rob. But when you go out and watch animals, you see that species that can rob, these individuals will sometimes visit legitimately. And so mutualism theory has not been able to address this challenge, this question yet. But this seems like a perfect place then to sort of switch our perspective and think about this instead of an evolution of mutualism question is just a foraging behavior question. And so that's what I did. And I use a behavior perspective to ask, why do these theoretical assumptions not always hold? And then when is it better to show behavioral plasticity versus constancy? And so this is part of a larger project during one of my postdocs, where we looked at nectar robbing from the bee's perspective. It has largely been looked at from the plant perspective, but now we're saying bee perspective, behavior, and look at this as a behavior. And this was in collaboration with Judy Bronstein and Becky Irwin. And we did this work up at the Rocky Mountain Biological Lab in Colorado in the US. This is an area very high up in elevation, and we were working in subalpine meadows, like the one you can see in the picture. And these meadows will have large patches of a single type of flowering plant, of these types of plants that have these long tubular corollas and get nectar robbed, and they're visited by diverse bee communities. So they're a really good system for studying nectar robbing. And we focused on 3 different plant species and 3 different bee species. So we have Corydalus cassiana, Mertensia ciliata, which is a bluebell, and Linaria vulgaris, which is actually a European species that's invasive in the US. And in all 3 cases, I've circled some robbing holes. If you peer closely, you may be able to see them. And then we have 3 bumblebee species, Bombus mixtus, Bombus flavifrons, and Bombus bifarius. So Bombus mixtus is capable of primary robbing, and the other 2 are not. They only secondary rob using existing holes. So what I did to measure the behavior of these bees was 2 things. So one, I did controlled experiments where I manipulated the food handling tactics that a bee was able to use on a given flower so limiting her to legitimate visitation or to nectar robbing, and then I measured her foraging efficiency. And to do this, I caught wild bees and cut flowers and brought them into a tent and tested the bees in there. And then we also, as part of the bigger project, did a bunch of field observations where we had a really great cruise of folks who went out to these patches of flowers, individually marked bees so we could follow individuals, and then would observe bees for a bout, so following her for as long as they could until she flies away and recording every single flower she went to, how she handled that flower, so that we could learn a bit about what these bees are doing in the field as well. And so the first thing I looked at is why this theoretical assumption that there's a cost to being a mutualism and that exploitation should always be favored, why we don't actually always see that. And so to do this, I brought bees into the tent, I made them visit a flower a certain way, and I measured their foraging efficiency with a specific tactic. And so here's our first case. This is Bombus mixtus on Critolus cassiana. And here we see that for these bees, it is more efficient to nectar rob. Here's their foraging efficiency, and it's higher when they're nectar robbing. And the reason for this is that these bees, they can't really push into these flowers and get into them legitimately. And so with legitimate visitation, they get very little nectar. With robbing, they get a lot more nectar. And so this makes robbing more efficient for these bees. So there are cases that are consistent with the assumption that there's a cost to legitimate visitation. And so for these bees, nectar robbing is more efficient. But we also see cases where it's more efficient for the bee to visit legitimately. And so this is Bombus flavifrons visiting our bluebell, Mertensia ciliata, and we see that there's higher foraging efficiency with legitimate visitation here. So contrary to the assumption from mutualism theory. And the reason for this is that it takes these bees a while to actually find the robbing holes and get into the flowers. Whereas they're very quick to just go to the flower opening, pop up to the flower, and feed that way. So it takes them a longer time to access the food through robbing, and this decreases their foraging efficiency. And then we also see a case where there's equal efficiency from legitimate visitation and nectar robbing. So this is Bombus bifarius on Linaria vulgaris. So we're seeing clearly it is not always higher. There's not always a higher payoff from nectar robbing. And in fact, we can have a lot of different relationships in the payoffs from robbing and legitimate visitation. So that's what I measured in the tent under very controlled conditions, but it also very nicely matches behaviors that we're seeing in the field. So if you look at what these bees are doing in the field, Bombus mixtus, when they're visiting Critolus cassiana, are almost always primary or secondary robbing. And this matches what I observed, that it's more efficient for them to rob than visit legitimately. Bombus flavifrons on Mertensia ciliata almost always visited legitimately, which again had the higher payoff in my experiment. And then Bombus bifarius on Linaria vulgaris showed they did a lot of both legitimate visitation and secondary robbing. Remember, the species cannot primary rob. And again, in my experiment, these two tactics had equal payoffs. So it does seem like these payoffs may be driving the behaviors that we see in the field. Which is what I say here. So it is quite possible that the reason that we see legitimate visitation by individuals that are capable of nectar robbing is that the payoff in that given circumstance is higher for legitimate visitation. And those circumstances are likely driven by things like the bees' adaptations for robbing, but also the specific plant species visiting. the bee-plant pair may be driving a lot of this. So then we're going to look at when it's better to show behavioral plasticity versus constancy. And so with a lot of different behaviors, you can sometimes see short-term fidelity in how an individual will handle food. Even if they're capable of handling food in more than one way, they may show this short-term constancy to one tactic or another. And this has some benefits. It can help avoid costs of increasing your predation to do more assessment, but also it has opportunity costs. So if you're sticking with one tactic, you may be not learning that another tactic is actually going to give you a higher payoff. So costs and benefits to tactic constancy. And I was interested in tactic constancy in the context of this nectar rubbing behavior. And so we have sort of three possible strategies here. We can have a bee that's constant to legitimate foraging. We can have a bee that's constant to nectar rubbing, or we can have a bee that's mixing tactics. And I'm going to be talking about tactic constancy just within a single foraging bout here. So first, let's look at what bees did in the field. So this is those three bee species that I talked about before, data from all of them combined on these flowers. And we see that in the field, we see a pretty high degree of constancy to single tactics. So on Corydalus cassiana, we see that the bees are mostly robbing, that's a sort of a mix of primary and secondary. On Mertensia, we see also they're primarily robbing. We do get some mixed bouts on the Mertensia, where we see robbing and legitimate visitation in one bout, on Mertensia, they're largely constant. Most of that constancy is to legitimate visitation, the blue. And then on Linaria vulgaris, very high amounts of constancy, most of them to legitimate visitation, but some also to nectar rubbing. So in the field, we're seeing a lot of constancy. And I looked at three different hypotheses. Maybe one tactic just has a higher payoff, and so that drives constancy. Another is maybe there's a cost to switching between tactics that could promote sticking with one tactic. And then the third is maybe there's competition for nectar that, and by, say, showing a strategy of always robbing, maybe you're able to get nectar that otherwise would be inaccessible, like those little bits up in the flower. So this could potentially drive constancy to robbing. So three different hypotheses. And and I looked at these largely through a set of experiments. So the first question is using the data that I already showed you, as well as the field data. So let's look at that. So we see that on Mertensia ciliata, where again, there was a lot of constancy, largely to legitimate visitation. These bees do have higher foraging efficiency with legitimate visitation than with nectar rubbing. So they may be constant here because it's the energetics work out that way, and it increases your fitness to just visit legitimately. But then we also see constancy to a single tactic when the two tactics are equally efficient. So these are the data from Linaria vulgaris. And so it's not just payoffs to a single tactic, clearly, that are driving constancy, because here the payoffs are the same for each tactic, but we're still seeing high levels of constancy, mostly to legitimate visitation. So it's not just payoffs. So now we're going to look at switching, whether there's a cost to switching tactics, basically. So what I did here was I set up an experiment where I had a bee first visit a flower in one way with one tactic, and then I forced her to switch in phase 2 to the other tactic, and I measured her foraging efficiency after that switch. So if there's a cost to switching, we would expect a drop in foraging efficiency. And as you can see here, we do not see that drop. So no matter which tactic the bees did first, their foraging efficiency is pretty much the same before and after I forced them to switch. And I did give them a little bit of warm up in the first phase so that they were foraging well when I started collecting data. So there's not a cost to switching. Also, competition is not driving constancy. So here we took a combination of the field observations where we brought in data on bee densities and how much bee activity there was in the patch at the time data was being collected, and also the nectar standing crop, how much nectar was available when we were doing the observations, but then also from 2 experiments where we manipulated competition levels and measured tactic constancy. And so these, in all 3 cases, there was no evidence that competition is driving constancy. So these are the data from one of the experiments with Bombus bifarius visiting Linaria vulgaris. And you can see that whether competition levels are low or high, there's pretty high levels of tactic constancy, and no significant impact of competition. So to wrap up this nectar robbing portion of the talk, there's assumption that there's a cost to cooperating is not always empirically supported. And in fact, we see variation in which food handling tactic has the higher payoff. And so by looking at this as a behavioral ecology question, we're able to gain valuable insights into the evolution of mutualisms. We also see high behavioral constancy. This is sometimes explained by tactic payoffs, but not always. And it's also not explained by nectar switching costs or competition. So why we're seeing this tactic constancy still is a big open question. And so by studying mutualisms through the lens of behavioral ecology, we're able to produce these novel insights about foraging decisions that bees are making and what the payoffs are from different foraging strategies and tactics. Okay, so now we're going to switch gears a little bit and talk about interactions with other foragers and how bees, both stingless bees and bumblebees, use social information about food availability. So information helps bees find food, and in general, in behavior, as an individual has more information, it's better able to match its current behavior to the current environment. And this, one of the types of information that we have is social information. And so social information is where an individual can learn about the environment by observing other individuals' interactions with the environment. So like this bee here is watching this other bee forage, for example, and maybe able to get some social information from that. And this contrasts with getting the information directly yourself and sampling yourself. And so bees seem pretty good at using social information. They're able to use it to learn which types of flowers are currently rewarding when they're starting to look for a new type of flower to feed on. So let's say the flowers they were feeding on, the nectar has all dried up, or that's a new bee just going out to forage for the first time. And then also they can learn which individual flowers are likely to have nectar or be empty using social information. And they'll use, for example, here's an example of visual social information use where this bee can use the presence of the other bee on a flower to learn about it. And we have really nice documentation that they use social information from conspecifics, whether that's nestmates or individuals of the same species but a different nest, and also from other bees. But something that I've noticed in my ecology research is that there are a whole lot of things that visit flowers. So these are just a couple of quick graphs. This is from an ecology study I did on canola, looking at the number of different insects in different groups that I found on canola flowers. And you can see there are a lot of bees, but there's also a bunch of like beetles and bugs, which are largely aphids, and a lot of different things on flowers. And then my graduate student, Rob Wiley, did some observations of wildflowers, some timed observations of wildflowers in North Texas. And he again finds a lot of different things are visiting flowers. So here he's looking at the number of different taxa on the flowers. And we see that, again, a lot of bees, but also a lot of things like beetles are visiting flowers. And so all these different insects may potentially be sources of social information for bees. It might not just be other bees that they're able to learn from. And the more information they have in general, potentially the more efficiently they can forage. And so we are looking at how bees use social information under ecologically realistic conditions where you have all these different flower visitors, and looking at which species provide social information, and then how do bees respond to diverse flower visitors. So I'm going to start with stingless bees, and this was the focus of my doctoral research. So stingless bees, they're eusocial. They live in a big colony with a queen. They're really important tropical pollinators, and you find them often in diverse communities. There's a report of over 70 species at one site. And they also show really high trait diversity. So looking at them, you can see like they look different, and they also have variation in things like where they nest. And they're very threatened by habitat loss as well. So they're great for studying behavior, and they also are important from a conservation standpoint. And these bees also are, pause the video. These bees also are interacting pretty frequently at flowers where they're competing for access to flowers and fighting over resources. So, and they can get, they're stingless, but they can actually be very aggressive. So I'm going to show you a couple of examples of that aggression here. And these are on artificial flowers that I made as part of an experiment that I'll show you some results from in the next slide. So let's look at these bees. So there are 4 individuals of the larger species and 1 of the smaller species. And the smaller bee, she's lunging, she's briefly biting these bees. She's trying to drive them off so that her nestmates can come and feed at this food source. And this second example is it's slowed down. It's the same larger species, but a more aggressive, smaller species. So they can be pretty mean to each other. And if you want to watch this video or use it in teaching, I use it in teaching. It is on YouTube, and here's the link. So, okay, so they beat each other up at flowers, and this can affect who's able to access specific flowers. So I used that array of flowers and showed that group foraging species are able to control individual rich resources. So these bees, they are all eusocial, but some forage just as individuals, while others will recruit large numbers of nestmates to a rich food source. And so it's these group foragers that are dominating. And if you remove the group foraging species, the solitary foraging individuals have increased feeding opportunities. And a lot of these group foraging species also will recruit nestmates to a rich food source using a pheromone trail. So a bee, she'll find a really nice resource, and then as she's going back to the nest, she'll make a sort of a trail by depositing little odor droplets. And she goes into the nest, sort of basically gets really excited, and so her nestmates will leave the nest, follow the trail, and this lets them very quickly get to this food source and start feeding from it. But these trails are out there in the environment, so they're susceptible to eavesdropping, to being used as social information by individuals from different colonies. And that can be a different colony of the same species or a different species. And today I'm focusing on heterospecific eavesdropping on these recruitment trails. And I'll be talking about what determines whether an individual is going to use the trail and follow it and go and quickly find that food source herself, maybe get her own nestmates to take it over and feed there, or if she's just going to ignore the trail and keep foraging as if she had not encountered it. And so as part of looking at this, I quantified the recruitment process. And so this is for a species called Trigona spinipes. And so I would have one individual come to an artificial feeder and then counted how many individuals were coming to the feeder and actively foraging there, as well as how much pheromone was being deposited right around the feeder. And so what you can see is you start out with just the one bee, and then the numbers of bees build up as she recruits. And when there's only one or a very small number of bees, there's not much pheromone right around this food source. But as the pheromone gets stronger, this food source, there's a lot more activity there. And so it's much more defended at that point, once the pheromone is stronger. And so then I also measured experimentally measured eavesdropping behavior. And so what I did was I would have one species, in this case, Trigona yelinata, that's my eavesdropping species, and then one species, here it's Trigona spinipes, the one I just showed you the recruitment pattern for. So the Trigona spinipes here, we're using their pheromone to see how Trigona yelinata respond to it. And so these were wild free-flying bees. I'd set out a pair of platforms, one of which would have the pheromone at a specific concentration and the other no odor. And I measured how many individuals went to the feeder that had the pheromone on it. And so what I'm showing you here is how Trigona yelinata responded to Trigona spinipes pheromone. So Trigona yelinata, that's the bee you saw throwing the other bee off the feeder. So they're a pretty dominant species. And they actually show a concentration-dependent response to the Trigona spinipes pheromone. So on the x-axis here, we have the pheromone concentration. The units are bee equivalents, which is the quantity of the gland that produces the pheromone from one bee is one bee equivalent. And then the y-axis is the average proportion of bees in one trial that landed on the feeder that had the pheromone. And so you can see when there's no pheromone, there's no side preference, that's good. When there's very little pheromone, probably they can't detect it. But then we get to this low concentration, this weak concentration that corresponds to just the start of that recruitment process, where there were not many Trigona spinipes on the food source. And here, the Trigona yelinata is attracted to this weak pheromone. But as the pheromone gets stronger over here, we see that they switch to actually avoiding this pheromone. And this corresponds, remember, with where you have a heavily defended food source. So what this suggested to me is that these bees are able to use both the chemical composition of the pheromone, which is species specific, but then also use the concentration to infer how much effort they're going to have to put into fighting to gain access to this food source. And if that inferred fight cost is too high, it's not worth it for them, is what this suggested to me. And so I really wanted to test this idea that inferred fight costs are affecting eavesdropping and driving eavesdropping decisions. And so I collaborated with Josh Graf-Ziven, who's an economist at UC San Diego, and we developed a decision analysis model. And so decision analysis models is a type of model typically used with human decisions to model the outcomes in terms of, often they're talking about money or health outcomes. We were talking about fitness outcomes, model the outcomes in response to a series of decisions. And so we developed this model for bee decision-making to say what's the impact on the colony energetics of attending to fight costs or sort of not attending to fight costs in making eavesdropping decisions. And we focused on the colony because for these bees, the colony is the unit of reproduction. And so I'm not going to walk you through the whole model. I'm going to jump straight into the results. And so we parameterized the model for 3 different species pairs. So first it is Trigona yelinata eavesdropping on Trigona spinipes, which are the data I just talked to you about. Then I also measured how Trigona spinipes responds to Trigona yelinata pheromone, and I found that they didn't care about it. Either way, they were equally likely to go to a feeder with and without pheromone. And then my graduate advisor, James Nye, had previously determined that Melipona rufiventris strongly avoids Trigona spinipes pheromone. So Melipona rufiventris is closely related to those large bees you saw in the video. Those were a different Melipona species. So we have 3 different cases with 3 different sort of responses by the bees. So we parameterized the model based on the biology of each of these species. And then we measured 2 different things to look at this. So overall, we found that the model does predict that Trigona yelinata and Melipona rufiventris should attend to fight costs, whereas Trigona spinipes should not, which matches their empirical behavior. So from the model output, we measured the attraction avoidance threshold, sort of the point at which an individual should switch from being attracted to the pheromone they're eavesdropping on to avoiding it. And so sort of indicates how long of a fight cost they're willing to endure in order to get access to this food source. And so for the Trigona yelinata, we found it's about an hour. It's sort of non-zero. And so that is reflected in this switch from attraction to avoidance. And then for Melipona rufiventris, we found the threshold is 0, indicating that they will never be attracted to the Trigona spinipes pheromone. And again, that's what we found in the field. And then the other thing we measured was basically how much of a cost is incurred if an individual makes the suboptimal decision. So how much, which sort of indicates the strength of selection to be attending to the information about fight cost. And you can see that both for the Trigona yelinata and the Melipona, there is a decent cost. This is about a day's worth of search effort. But for the Trigona spinipes, there's no cost to making the suboptimal decision, which is probably why we saw 0 preference by these bees. So overall, the model shows that using a decision rule of attending to fight costs and basing your eavesdropping decisions on that fight cost likely is increasing colony fitness, if it has any impact on fitness at all. So that was really exciting. So overall, with the stingless bees, we saw that they're able to infer fight costs from social information using both the chemical composition that's species specific, but then also the concentration. And they respond in a way that minimizes their costs. And this has implications for the evolution of communication. So when we learn about evolution of communication in the presence of eavesdroppers, typically what we hear is that eavesdroppers should be selecting for non-conspicuous signals, and signalers should make their signals less conspicuous in the presence of eavesdroppers, because eavesdroppers can more easily use the information and potentially harm the signaler. But a lot of this comes out of the predator-prey context. And here we were looking at eavesdropping in the context of competitors for food sources. And what we see is that here, eavesdropping may actually be selecting for conspicuous signals. So if a colony is able to say like, hey, produce a stronger pheromone concentration that indicates like this resource is already really occupied, just like don't bother because we're going to make you fight for it, of course, heavily anthropomorphizing here, then there can be a benefit to making your recruitment trail more conspicuous. And so then there also may be some ecological implications of this this eavesdropping and of social information use in general, I think, in this context. And so I'm just going to very briefly give you a little peek at the ecological side of my research. So I collaborated with Barry Brosi, who had data on stingless bee species present in pastures in Costa Rica that were surrounded by varying amounts of forest habitat. And then I had a species traits database, so we combined these and to look at whether specific species traits affect susceptibility to forest loss. And we found that they did. And sort of the big takeaway here is that foraging dynamics, such as these dominance interactions, and probably also movements among flowers that are driven by things like eavesdropping, these foraging dynamics affect perceived diet breadth that us people see, and this diet breadth affects susceptibility to forest loss. And so it's quite possible that social information use is one of the factors that's driving which species are lost from communities under habitat change. Okay, so now we're gonna switch over to looking at bumblebees and their social information use. The stingless bees, we're looking at social information from signals. Bumblebees, it's just cues. So we call that spying instead. And so this is work my lab is currently doing. You saw this graph before. Remember, a lot of different things visit flowers. And so we were really interested in whether bees are able to use social information from other types of flower visitors, such as these nectaring predatory insects. to learn about flower availability. And so that's what we're currently testing. And we're looking at this with lady beetles as our nectaring predator. And so we're looking at how bumblebees use social information from other bumblebees versus from lady beetles. And I should note that these lady beetles, they're not predators of bumblebees. They eat things like aphids and smaller insects. And so what we do is we buy commercial bumblebee colonies. So this is the inside of a colony here, and we set them up in the lab. Here's the colony under here. The bees fly in this little flight arena, and then we have an array of artificial flowers that we put out that we can have with or without nectar, and also put different types of social information on. And so we're looking at visual social information in the data I'm about to show you, where we took dead bees and lady beetles and we put them on pins and we removed all the odors from them. And so we can put these little pinned insects on a flower to experimentally control social information. And this is being led by Marie Muniz, who was an undergrad in the lab. And we asked two questions. So first, do the bees innately recognize lady beetles as sources of social information? And then are they able to learn through experience that that lady beetles can be information about food availability? So for looking for recognition, what we do, we put out two flowers, one with, one without a model or demonstrator pinned insect on it. And we look at which of those two feeders our bee chooses. And so we see that, as many other studies have found, or several other studies actually have found, when there's a bumblebee demonstrator, the bees that are using the social information are strongly attracted to the feeder that has the bumblebee on it. And I should note that the bees that went into these experiments, on a daily basis, they're feeding on a feeder that's ad libitum in the presence of other bumblebees. And so this is sort of the behavior we expect with a bumblebee demonstrator. But then they did not use the lady beetles as social information. So they did not have a preference for or against a feeder that had a lady beetle on it. So then we looked at whether they can learn to use lady beetles as sources of social information. So here we gave them multiple foraging bouts to learn the association between a bumblebee demonstrator or lady beetle demonstrator and food. And then we tested whether they had learned that association. And so we have 4 different treatments here. So our demonstrator can be a bumblebee, or it can be a lady beetle. And then the flowers that have demonstrators on them either have food or are empty, and it's the other types of flowers that don't have demonstrators that have food. And here's what we found. So when the bumblebee is the demonstrator, the bees do easily learn that a bumblebee indicates the presence of food or the absence of food. And when we're testing their learning, they strongly prefer those feeders. They can also learn that a lady beetle indicates the presence of food. So they can learn to use lady beetles as social information. But when the lady beetles were present on empty flowers that did not have, they actually had water in them, but they did not have sugar water, no reward. The bees did not learn to associate lady beetles with empty flowers. So it's possible that maybe learning these insects that look very different from bees, maybe this takes more time, or maybe there's some other cognitive constraint on learning, on using these nectaring predators as social information. But they can, at least in some circumstances, they can do it. So to wrap up this portion of the talk, to wrap up the entire talk, we're getting to the end. We see that behavioral ecology can help us better understand mutualisms. We can explicitly test these assumptions that have long been held about payoffs from different foraging tactics. But it's still a really big open question about what's driving tactic constancy, because it's not just payoffs, and there's not a measurable switching cost. So big question there. We also see that many different species potentially are providing social information, including species that are in different ecological functional groups from the individuals that are using the social information. And so as long as there's a shared resource, maybe bees are capable of using a whole lot of different insects as social information. And we also see that there's some really interesting decision rules that these bees are using. So they can avoid fight costs, for example. And they're maybe they can learn from nectaring predators, but it's possible that they're treating them differently than bees when they're using social information, which is something my lab will be looking into the future. And I said early on that my lab integrates behavior and ecology. And so these interactions I've been talking about, these behaviors that I've been talking about, they also can scale up to regulate ecological processes. So there's some really exciting questions in that area as well. So we saw, for example, that these behaviors can affect how communities are disassembling and changing as habitat is lost. And it's also strongly likely that these foraging behaviors, they're affecting how bees are moving around flowers, which specific flowers they go to. And so they're likely altering pollination and plant reproduction, which I think is a big open question and an area that my lab is moving towards. And of course, this work would not have been possible without many, many people. And I would be happy to take questions. Thank you.
Great. Thanks so much, Elinor. Very, very great talk. Very interesting. And we actually already have a ton of questions in the chat, which is great to see. So I'll start with our first question from William Russell, who in his YouTube name also says he is looking for a PhD. So his question is, could the answer to the mutualism theory question be related to exploration and play? And do bees generally like to try out different behaviors and strategies, even if they have a valid solution?
So the data I was collecting are not set up to fully address that. So certainly it is possible. These bees are, they're under selection to be foraging efficiently. There's predation, for example, by spiders and birds. They also, for some of these species, because they're up in the mountains, they have a relatively short foraging period. So some species may only be active for a month or two. And so we do think there's pretty strong selection for them to be foraging efficiently. And so they will probably, less experienced foragers are probably trying out different food handling tactics. All the bees that I used, we caught them while they were foraging and saw that they knew what they were doing basically. So we were only testing bees that with reason to think we're experienced foragers. But certainly early development of these behaviors is another question. And there's a little bit of research on that. So Jesse Barker looked at this in the lab, sort of the development of robbing behavior. But these bees, they do silly things like hang by one foot off a flower, but they're pretty to anthropomorphize. They're pretty businesslike when they get to flowers. They're there to get their food.
Great, awesome, thank you. So our next question is from Natasha Rossi. She has kind of two back to back. The first one is, how did you measure calorie intake? And I think this was from sort of one of your first experiments you talked about.
Yeah, so with the nectar robbing, so what we did, we knew we needed to standardize sort of the nectar volume in the flowers. And so what we decided to do is we very carefully emptied out all the nectar that was present in the flowers and then use a narrow syringe to fill the flowers with a specific volume of a known concentration of sugar solution. And so we knew how much was in there. And then after the experiment, we measured how much remained. And from that, we could calculate how much sugar the bee had consumed in her visit to the flower. And we also recorded how many visits she made to each flower during the course of the experiment.
Okay, great. And her second question there is, did you also look at the same flower species for different bee species?
So this is the nectar robbing. So I looked at those 4 combinations that I told you about, and the only one I did not talk about was Bombus mixtus visiting Mertensia ciliata. And I've got this data somewhere down here. And so, yeah, so what we see here, so when Bombus flavifrons visited Mertensia ciliata, they had a higher payoff from legitimate visitation. When Bombus mixtus visited, they had a higher payoff from secondary robbing than from legitimate visitation. But in the field, they showed a lot of legitimate visitation. And so what, but robbed flowers often have less nectar, or flowers that already have robbing holes in them, which is what you're using for secondary robbing, often have less nectar in them. And so when you take that into account, this even though in a tent where everything's equal, they have higher efficiency from secondary robbing. They actually, I did some calculations that estimate that in the field where the robbed flowers are largely empty, they're actually going to have a higher payoff from legitimate visitation. So it does match what they see in the field. So that was the one case where we looked at two different bee species visiting the same flower species. And we do see that we have evidence here that the story is not quite as straightforward, but we do have evidence that payoffs are likely also driving the behavior in this situation, even though it has to sort of take into account nectar standing crop as well.
A lot of things to keep in mind, and all the mixes and different experiments you can do.
Yeah.
And so actually there was a third question from Natasha as well. Did you look at the effect of flower morphology on bee tactics?
So we did not measure that. That was not part of this study that I did, but we do. So it clearly has an effect. So for example, the Bombus mixtus, they cannot push open the Britillus cassiana, that was the whitish purplish flower. Some of the larger bumblebee species can, and so are able to visit legitimately, but the smaller bumblebee species, they can't even get in legitimately. So legitimate visitation is not really an option for them. I also saw sort of anecdotally the Mertensia, the bluebell. My second year there, the flowers seemed to have longer and narrower corollas, and the bees could not visit legitimately and get any nectar in a lot of those flowers. So the flower morphology definitely is affecting what tactics are available to a bee and what the payoffs are going to be.
Good, thanks. So going back to William Russell, he had another question. When you forced a bee to switch tactics, did they maintain consistency to the new tactic even if the original tactic became available again?
So I switched one time. I did not let them switch another time, and I was very strictly controlling what tactic they were able to use. So for example, all of the flowers would be glued shut, or all of the tops of the corollas would have little straws over them that prevented them from robbing. So I did not do another switch.
Okay. And he also commented, I wonder if individual bees have a preferred tactic. I don't know if that would be a question, more so just kind of like a curious, because I guess it's hard to tell what each individual bee is thinking, but it is interesting.
Yeah, so it's quite possible. Anecdotally, the Bombus mixtus, they really like to nectar rob, and they have mandibular teeth, so they're able to bite these holes. But yeah, we weren't able to look at that in my experiment or set of experiments.
Okay. And then just another question from the chat, Prabhayan Ghosh. How are the plants adapting to these nectar robbers? Do they does making a hole in a flower decrease their survivability, or can some other insects and organisms then get inside the flower?
Yeah, so for a more detailed look at that, Becky Irwin and some colleagues have a review from 2010 of nectar robbing. It'd be a great place to read about that. But so one of the sort of the evolution of mutualisms, ecology of mutualisms body of literature has been something they've been unable to answer is, well, if under this assumption that there's always a cost to legitimate visitation, so bees should always rob when they can, why are these plants doing fine in terms of their reproduction? Because there are many systems with nectar robbing where the robbing does not negatively impact the plant reproduction. There are some where it does, but there are also some where it does not. And so quite possibly there's just enough legitimate visitation that the plants are getting pollinated as well as nectar robbed.
Interesting. Yeah, I guess a whole other area of research into the plant survivability.
Yeah.
And then Natasha Rossi had another question. Do you know how much pheromone is naturally deposited on a trail by one individual?
So let's see. Yeah, so I have some rough estimations of that. So I was counting the number. So in the graph I showed you, I counted the number of pheromones deposited. They do deposit more heavily around the food source. And so I took advantage of that. I put like a big piece of paper up, and they would love to mark on that piece of paper. And so it was easy for me to see how many markings they were doing. But then I did a smaller number of trials of the eavesdropping behavior with actual odor marks. And so when there were just 4 odor marks, the Trigonia elinata were attracted to 4 of the Trigona spinipes odor marks. So that's about the 0.075 bee equivalent. So I don't know the exact numbers, but this is roughly the amount. When there were many odor marks, then we see the avoidance. At 2 odor marks, it looked like they couldn't really detect 2 odor marks. So it is really just, a fairly small number is sort of the weak concentration. But then once you get the buildup, the Trigonia elinata, they switch to avoidance. And so these are actual counts here. Sort of inferred from both that result I just showed you and the model.
Awesome, thanks. And we are at time, but we'll wrap up with the final question from Natasha again. How do you remove odors from a corpse?
So we dunk the bees once they're dead, of course, we dunk them in hexane several times, and that strips off all the odors. And then we keep them in a desiccator so that they're kept nice and dry and not going to start molding or anything like that. And we're very careful in how we handle them. And we handle them the day we're using them in trials, we handle them only with forceps. So we're not getting our fingers all over them either. Because bees can also use chemical social information. And so we really wanted to avoid that.
Great. So that was all the questions from the chat. We also have some comments coming in saying, thanks for a super interesting presentation. Thank you for answering my question. And then those little clapping emojis. So thank you again, Elinor, for kicking us off to a great 2024. Thank you to everyone who attended and was active in the chat. Our next seminar will be at the end of April, on April 30th. So just keep an eye out on our social media for more information on that. So thank you again, everyone, and we'll see you next time.
And thank you for having me.
Thank you.